And now I'm going to shut up and turn it over to Rashmi to tell us what happens next.
Well, first of all, thank you so much, all of you, for being here and taking the time. And, we are really grateful. There's a lot of patients as well listening who'll have access to the videos later. And we think the session is going to help doctors and patients. In terms of what comes next, it's the working sessions. And the goal of the working session today was to really kind of digest the morning 2 sessions, which is the diagnostic criteria session and the IL18 trial session. I have set up a Google Doc, and I can, Luciana and I can be made the note takers. And I invite, those who were in the morning sessions and others who are here and interested to join in. We don't really have a real format for what's going to happen. So we are going to kind of discover along the way. Scott, Alexi, Fabrizio, Grant, you guys want to take over?
Is Pete here? I can see Peter Nigovic. Is he here? Can he? Here he is. I see.
For about another 11 minutes.
I couldn't see you. I think you are the best. I don't want to put you on the spot, but I will. I think you're the best person to wrap up on the diagnostic part of it. If Scott, Alexi, would you agree that Peter is the best person?
Absolutely. I thought that I was very compelled by the basket trial argument. But I think that what I learned from this session from our session was that we should be thinking about new onset disease and prevalent disease a little differently, in the sense of criteria for entry in study trials. I am still compelled by Bas Vastert's work in Utrecht that most patients like this have respond well to one course, and it's those who don't that we then need to work on in the subsets. But since we don't at this point have a way to tell who's who, in a way, therapeutics is the way to try that. I like that idea. I think that I think arthritis should be dead. I just don't think we should require arthritis at all. I think the data that it is an artifact of the fact that, as Ed Behrens was saying in the comments, that this had been at some point lumped in with the juvenile arthritis group, and otherwise nobody would think it was necessary. It was just one of the many features of the Still's syndrome. So I think that should be dead. And I think the adult pediatric distinction should be dead. I just think those should be non-issues, more generally in rheumatology, but I think in particular in this disease. Then I was struck by Nikolai's point that what the FDA requires from us is, first of all, for us as content experts to make the decision. And that means coming together in some kind of consensus. And then that when we come together to design studies, we need to have measurable outcomes to assess efficacy, including ways that the average clinician, as I think his term was, can identify a population of patients. And I think IL18 is such a criterion, as long as we get the right test that works well across centers, I think. So I would say those are my.
Could you clarify this statement? So when you say IL18 is a criteria for what? For
so I think
choosing the right dose, or as a efficacy?
No, I think as we pick a group of patients that is identifiable by the average clinician for entry into a trial. So you could say all the patients with these criteria received this trial, A or B, and they did these outcomes. So we have to make sure that this entry criterion is not just they look good to me. They have to be some kind of list of things. But as Scott was poking at us, saying that we're doing the old school thing of trying to use clinical criteria when we should be using the old fuddy-duddy thing, and we should be doing the laboratory, some kind of biologic index. So I think that one thing that we should consider is as to use biomarkers that seem to have incredibly strong predictive, I would say, discriminant value. So the IL-18 is almost like a CCP test in rheumatoid arthritis. Like if you've got it, you are much more certain that you have the right diagnosis than if you don't. Not everybody, but the vast majority, I think Bas said all but 3 to 5% had very high levels, over 1,000, not over Scott's 20,000. So I think that we can use that as an entry criteria and study those populations.
So one of the things that could be useful in a trial is to show that you target a certain molecule, let's say, and you see some clinical changes, and those clinical changes parallel the changes in the level of that biomarker, right? As an example, in MAS clinical trials, we are looking at the CXCL9 levels and show that the clinical improvement parallels the decrease in the levels of CXCL9. That's a bit trickier with IL-18 because total IL-18 remains high for a long period of time. If you go back to the clinical trial of canakinumab, in fact, the levels started going down even in those patients who responded beautifully on day 3. The levels started going down by month, I would say 7, 8, 9. So, but that's total IL-18. And the question I have to Scott, if we focus on free IL-18, would that be helpful?
Yeah, I think so. So this is a test that I've only been doing in my lab for the last couple of months, and we've only just begun to sort of thaw out the samples from the bigger cohorts to see how discriminative it is. I think that there is. I think detectable free IL-18 is fairly specific. I think that there's a sensitivity issue that there may be patients who benefit in whom we may not pick up on the free IL-18 assay. But for a sort of proof of concept trial, I think that that's probably okay. The issue is, the free IL-18 assay, just doesn't have the wealth of data behind it. And so it's the kind of thing that certainly, especially if, as Nikolai pointed out, having a real companion diagnostic, not just something that's run in a CLIA lab, is at least looked upon favorably, if not sort of a requirement. Sets up an issue for, is this a situation where if the drug companies are not interested in developing this as an FDA-approved test, is there sort of a venture philanthropy type model where, a patient organization starts a company, the company develops the test then gets sold to hospitals across the US. And that can be true for total and or free IL-18. I can see it happening for both.
Can I comment on this? I'm afraid we are maybe making things more complicated than needed here. One, we have to, I don't know whether you've seen in the chat the comments from Nikolai, which I totally agree with. Using a complicated biomarker or even a biomarker in general may decrease the generalizability of the results of the clinical trial. And if I may add to the very polite comment from Nikolai, is that I may actually be very difficult to write a label for such a drug. I don't know if I'm stepping away from my field, but I feel it's going to be very complicated, and I see Nikolai's smiling. The other thing is that I don't think, Scott, we need to go into free IL-18. I think the IL-18 assay, total IL-18, is very, very related to free IL-18 somehow. And it's very difficult, as you pointed out in several sets of data. This is the take-home message. If I'm not mistaken, then total IL-18 is fine. And then there is a system with an FDA-validated tool that can be used to measure IL-18 as a point-of-care system. Unfortunately, this is not EMEA validated. So I think we are very close to that. And I think we don't have the luxury to spend 2 years to develop a companion diagnostic the way you suggested. I think we don't have this luxury. I think we have to do it essentially now, and we have to be ready to go to the regulatory with the available methodologies that are very reproducible among different labs. We all have similar findings, and we know that high IL18, we can have a consensus on the cutoff. We can get data, but high IL18, 20,000 preferred by Scott, a few thousands, maybe less by us, but that's okay. We can find a consensus on a level that is reasonably picking up the great majority of these patients. And I don't think we should make it more complicated for us, for the sake of time.
Yeah, I just to jump on that, I think you're right. I mean, I have measured IL-18s above 20,000 in quite a few non-systemic JIA patients. They were all of them quite recognizable as not systemic JIA and would be excludable. These are horrendous viral infections. These are malignancy-associated HLH or EBV HLH or both. So patients that I'm not too worried are going to slip through the cracks of an inclusion-exclusion criterion.
Right. And then you're not using IL-18 alone as the entry criteria. You're using clinical diagnosing of this disease, whatever we call it, and IL-18. And we will probably identify the difficult ones.
But I think, the maybe most important but absent voice here is from industry. When I speak to folks in industry, I get the impression that they don't want to challenge these paradigms of diagnosis because the FDA doesn't like it. But here we have our heroic FDA representative telling us, you're the experts, you tell us how you want the sort of average clinicians in your field to be diagnosing these diseases. And so, I think we need to bring that back to the industry partners that we have to say, these different inclusion are not hard stops in terms of getting an indication. Any pushback on that, Dr. Love?
I'm fine. I think we need to move that direction.
Again, I don't think this is a call for me, or I think it's not necessarily for FDA to address.
Right, but you are being scapegoated whether you like it or not. Dr. Lovell has his hand up.
Yeah, I think we need to be careful here. Scott, your presentation about IL-18 was thought-provoking to think about expanding the spectrum of IL-18 diseases that could perhaps be studied in a basket trial, but there's also a large, people need to be able to diagnose and treat systemic Still's disease across the age spectrum, and that's I think we do have clinical criteria by which that could be done, so that physicians could diagnose the disease and use available approved treatments in early disease that we can't now because of the ILAC criteria. But I think what Peter talked about was coming up with a unified diagnosis of SJIA, Still's disease, whatever you would call it, based on clinical and laboratory, readily available laboratory measures. So I think there's merit in that, with the understanding that it is a spectrum disease, and there will be patients within that spectrum disease. That disease is primarily driven by IL-18, and we need to think about them treatment-wise different. But the reality is that 70–80% of the patients we call systemic JIA adult onset Stills are very well treated with IL-1 and IL-6 treatment. So I think we don't want to throw the whole group of patients in disarray here. We have a mechanism that we could, I think, easily adapt by just coming up with different criteria for diagnosis that would allow us to treat early and effectively the 70, 80%, that wouldn't exclude these patients that we're concerned about that have resistant disease. And then you can identify them more specifically by using markedly elevated IL18 and include these other monogenic patients in the same basket trial. So I think you get at what you need to try to identify these resistant patients with perhaps a different mechanism disease, but by using these criteria and then use IL18 markedly elevated as a entrance criteria for looking at a subset of patients that needs a different treatment because they have a different pathogenesis. So I don't think we're at the point where we ought to kind of roll over and characterize these diseases as exclusive by IL18 levels.
Yeah, I mean, I think
Nikolai,
any trial that includes those patients is kind of going to de facto end up with refractory patients anyway, because patients are probably going to be loath to start a clinical trial if they haven't tried an IL-1 or 6 inhibitor.
Well, I think we need to get beyond the clinical trial thing and talk about clinical diagnosis and clinical care on a routine basis, which is where we were coming from in these discussions about coming up with new criteria to enhance ILAR, because we need to think about regular clinical care for clinical patients as the primary goal for diagnostic criteria. And then for those patients who don't respond to standard therapy, try them on experimental therapy, unless we can come up with a better way to identify those patients de novo at the onset of their disease. And maybe we ought to be doing IL-18 assays at the onset as part of our initial evaluation of patients to select out those ones that are less likely to respond. But I don't know that we're there yet with new onset patients.
We started doing that in Cincinnati. Now it's a part of the initial evaluation, which will always look at IL-18 levels right away. But I agree with you. There are sort of two separate issues. One issue, the reason with this we need to adjust this criteria is to be able to start the treatment, just sort of conventional regular treatment for patients with systemic JIA as early as possible. But Bas presented this morning that clearly, they started treatment right away with whether those patients have arthritis or not. The outcome looks fantastic, right? So totally better than what we are seeing here. So the one issue is to modify this criteria to enable us to start the treatment as early as possible. And then there's a completely different issue related to sort of improve the eligibility of these patients into clinical trials. So there are very, very separate issues, and then you pointed it very, very correctly, I think.
You are right, Alexei. We should really try to change the getting rid of arthritis. I'm quoting Peter. Get rid of the word arthritis in the criteria. We have to find the best way to do that. In the meantime, I guess all the experts agree that this should be done and must be done. We have to find a way to reach consensus and doing methodology in a manner that is methodologically proper. Okay.
Nikolai, your question about MAS, we will discuss it tomorrow, so we will address that separately.
And the other thing I was saying,
I acknowledge that. I just noticed that there were several examples with high IL-18 levels, for example, that include MAS, and I think I'll leave it again to you guys, since you're the experts, of whether to conflate these two or just look at them as separate entities.
I think we should look at them as separate entities. We know that SJIA patients develop MAS. It's not like, there is not a straight line. Sometimes there are patients in between, but the point is that we have an issue with early diagnosis of SJIA, and we need to change and accept to get rid of arthritis, and I think we did. And this is good for clinical practice in addition to criteria for clinical trial inclusion and labeling, if I may say so. Second, we have an issue in defining the high IL18 spectrum of the disease, which most often, as Scott said, is refractory SJIA, are the bad patients, the difficult to treat patients. And we have to find a way to use the present state of knowledge to allow inclusion in clinical trials for this patient. And we have the third issue, which you just brought up, which is MAS, okay, which is trials for MAS, which are different for SJIA. And this is a completely different issue, as you well know, all of us know, and it's going to be addressed tomorrow.
But coming back to defining this sort of clinical population that we're talking about here, there are patients who present with some features of systemic JIA. It's sort of not classic, but still, for at least for coding purposes, I am comfortable calling them systemic juvenile idiopathic arthritis, although sort of I'm impressed with super high levels of IL18 and some other sort of features that may not necessarily fit with classic SJIA. But and those patients are in the same spectrum, but I'm not even comfortable using the word SJIA, but I still believe it's the same spectrum. Sort of, what patients are we talking about here? We
should come up with a name for the thing. I call it this disease because I hate to use SJIA, and I like the fact that arthritis is something that, you know, it's a leftover from the JIA, the chronic, the JCA and the JRA era. But we shouldn't call it SJIA. The adults did it better, I think. Adult onset still's disease is what by Water described, as Peter pointed out this morning. So we have to come up with a name. And the regulatory, some of the label states Still's disease, including adult onset SJIA. So we have to come up with a name. Yes.
So we sort of asked this before. Nikolai, what would you think if someone came to you at a pre-IND meeting and said, we would like to do a trial in Still's disease with a lower age cutoff of 12 months using the Yamaguchi criteria?
So what exactly is your question to me?
I mean, could we just call it Still's disease? Could we just use the Still's criteria? Is that going to be a problem, even though the criteria were developed in adults? But if the experts are telling you we're fine with these criteria for this population of children as well?
So I think we as an agency have considered Still's disease sort of as an umbrella diagnosis with the spectrum of different manifestations, including SJA and adult onset Still's disease. So I think that could certainly be a consideration. I mean, I think Peter made already a pretty clear argument about the differences in nomenclature and how these may need to be reconsidered. And I think we have taken up the discussion from the JIA workshop a couple of years ago and considered this argument ultimately to approve canakinumab for Still's disease with data in adults and pediatric patients.
Yeah.
Well, there was another interesting piece of data that you used. Remember, there was a gene expression study showed that, in fact, in terms of distinguishing gene expression profiles, there was no difference between the patients with adult onset Still's disease and systemic JIA. And the response to canakinumab, again, at the level of gene expression was absolutely identical.
Right, and I think, again, this was an argument made at the workshop discussion. I think the differences in nomenclature and classification between adults and pediatric patients has made it very hard for us in general. And I'm glad that the pediatric community has taken the lead to address some of these. I know that there are still outstanding questions, but I think for several of these diseases, we have considered the scientific evidence, including the polyarticular JIA and rheumatoid arthritis, psoriatic arthritis in adults and pediatric patients, also adult onset Still's disease and SJIA. So we have considered the feedback that we received from the workshop and the scientific discussions in the community. And I think where we feel comfortable with these arguments and with the science, we have taken already actions. So I think to your question, Scott, I think bottom line is if the population can be defined, and if that includes adults and pediatrics, that's even better. That should be acceptable. I think where we may have more troubles is where you have a disease in adults, whether these patients should be, or the adults should be studied first before you move to the pediatric patients. I think in the case of SJIA, it has been the other way around, where SJIA patients have been more easily studyable than adults. I don't know if that helps or answers your question.
I just want to point out there's several parents who've spoken up, and this is something that, if you want to be patient-centered, it's worth listening to, that they agree with you, Nikolai, that we all prefer actually stills and having the same criteria, and age of onset not being a reason to define the diseases differently.
Yeah, I mean, we also, this was something that I personally haven't thought about before the workshop, is sort of the transition of patient care from pediatric to adult rheumatologist or to adult patient care, where if you have differences in nomenclature, that can actually hamper the transition and the care. So the more alignment there is, which should be based on the science, of course, between adult and pediatric indications and diseases, should be pursued as much as possible by the community, and so it would make our job easier also to even require pediatric studies in certain cases.
I completely agree with you. One of the biggest things as kids grow older is that, and I think Kari, my colleague in the foundation, can speak up about this, whose kid is just going to go to college. And the biggest issue with transition is actually the fact that the diseases are named differently, defined differently. And, it's so difficult to get the same set of medications. Sometimes you go to an adult, rheumatologist, and you have to start with methotrexate. So that is a issue.
So we have to use the same name. That's pretty clear to me. And the FDA and the EMEA have both used Still's disease.
Yes, I certainly agree with using the same name. I think that makes complete sense. As far as figuring out which criteria to use, would it be helpful to, test the criteria on a large population of systemic JIA patients, for example, in the CARA registry or other registries that exist, to make sure that they're applicable to most of the patients that physicians do diagnose with Still's disease or systemic JIA?
Is that a question, Yuki? I think you would use this. That is not a question. It looks to me like it is a statement. Yes, it would be useful to do it. If I may answer yes, but it doesn't make sense that I do answer. What I really like to point out is that it's not the applicability of the criteria per se, it's the time that is needed for a patient to meet those criteria. So it's really, and time is an issue, as I said this morning. So I think it's the time. It's not just the applicability, it's the time it takes to fit the criteria. So I was thinking of doing it multicentrically among us in various ways, but I guess we have to look at the patients that have been seen from their disease onset. I mean, ideally by day 0, which can't happen, but maybe it's week 2, okay? In referral centers that are used to make the diagnosis, they can monitor and report the flow of the symptoms over time in the first few weeks, because this is what matters, weeks.
I agree, Fabrizio, but we can't be pediatric centric. That was what hampered us in the Printo. We had you have to
I-
include adult
I
onset
totally
patients.
agree. I totally agree, Dan. We can't be
Yeah.
pediatric center. This has to be done with adults. Absolutely.
Yeah, so that's what hampered us in Printos. We didn't have adult colleagues there. So it seems like your European effort is combining both adult and pediatric
Yeah, but
rheumatic disease specialists to come up with test cases in which you look at the various criteria. I was impressed in the small number of patients that were presented before, how the Yamaguchi criteria did not work very well for the patients you had. And so I think we need to be,
yeah, it's
this certainly needs to be data driven using adult and pediatric cases to see if we use existing criteria or some modification of existing criteria when we look across the age group.
Angelica has her hand up.
Yeah, hello. I am an adult patient with SJIA, and when I was transferred to like adult rheumatology, then it's a lot less like they don't know that it's still then that but they don't think that adult onset Still's disease and systemic JIA is the same. So they have different treatments, and they have like, I live in Norway, and there is one hospital that has a lot of competition and knowledge, and they know a lot, but they primarily only have patients who has adult Still's disease, and they don't see systemic children or adult with systemic arthritis, because I think the name makes it sound so different that they don't connect that it's kind of just the same disease, just different age of onset. So I really think it's about time to get the same name.
You're very right. There's nothing else we can say. It's
thank
really,
you.
it's too late. It's not about time. It's late, but we have to do it.
And we've actually had issues even in academic centers where patients who have Still's disease can't find an adult rheumatologist who will see them. We've had adult rheumatologists say, you know, I don't manage Still's disease. You've got to find someone else. So
The point is in adult, it is much rarer than in children. So if you look at the curve, or it's probably significant, the incidence rate is significantly lower in adults. And this makes it very complicated to do trial in adults first and then go to children. I think this population should be investigated all together. In age 12 months and over should be fine.
And I see that there was a question to me. So I'll try to take on this. So the question is, there are patients diagnosed as children, now young adults, who were excluded from some IL18 trials because they were diagnosed as children. If the name were the same, that would not happen. I don't know about the specific case or cases. If the diagnosis is the same between adults and children, generally we would want to have some evidence of efficacy, even preliminary evidence of efficacy in adults, to make sure that there is a prospect for direct benefit for pediatric patients. I think that's kind of a basic principle that we have, unless this is clearly a pediatric disease where there are no adults to study. So one reason to exclude children from some studies is that we, again, pediatric patients are a vulnerable population, and we cannot assume that the drug is effective. And I think we'll touch on this maybe during tomorrow's discussion. But until we have some evidence of direct benefit or prospect of direct benefit, it's very hard for us to justify exposing pediatric patients. That comes down also to some ethical concerns. Once we have that evidence, a study could be amended or expanded, or to include pediatric patients and adolescents. And we have actually endorsed this approach more broadly for pediatric indications, where we would actually encourage enrollment of adolescents at least. But again, that's a point of a different discussion. And again, I'm not sure whether that answers the question, but
I'm afraid I see a danger coming that it's a double-edged sword. So if we assume that if we take the position that the disease is the same, then we would require a trial in adult onset disease before treating children, and this would be extremely dangerous, and we lose a lot of time. The disease is much more frequent in children. There is an adult counterpart which is significantly less frequent, markedly less frequent. On the other hand, I see the point of, just predefining the age of the population, and the adolescents are as vulnerable as a 20-year-old. They're probably less vulnerable than a 40-year-old. I'm not sure they are. Probably the children are less vulnerable than adults. They take a lot of abuse, much more than adults in general. But one can see rather starting from adolescents and up, and then opening to lower ages. But I don't want to think that we have to do a trial in adult onset disease before getting into children, because this would take too much time.
Right. I mean, I think I just wanted to clarify that this is kind of a general principle, but in diseases where the disease is predominantly pediatric disease, then, I don't think we can justify delaying studying or access to drugs to pediatric patients. And in general, SJIA studies have been done way before any adult studies, just because it's logistically much more difficult to do the adult studies. And we have recognized this for, stills in general. But for diseases, other diseases, for example, lupus or some other diseases where you have, sufficient adult population, this would be a principle that we would follow.
And would you consider enrolling adolescents 12 and up into an adult study? Because, these diseases, this is not SJIA clearly, but this is an approach that would make the life of the adolescents much, much better.
Again, if the study is in adults and we have some evidence that a drug might be effective, essentially to address this prospect of direct benefit, this is under the regulations, and we have to abide by these regulations. We would definitely encourage enrollment or inclusion of adolescents. What we've heard so far is that there are a lot of logistical challenges, and I know that some people may have stronger opinions about that, but from regulatory perspective, the dosing in adolescents wouldn't be that much different from an 18 or 20-year-old. So dosing per se is probably well informed in adolescents to say, you can go ahead and dose these patients. For younger patients, that might be more difficult without additional PK, safety, and efficacy information. But that information could come from adolescents in patients, in trials that include, in adult trials. Again, we have raised this as one way of trying to expedite pediatric drug development, which could also help with or streamline the extrapolation later on for the smaller kids, which wouldn't require, the randomized controlled trials that were done in the older patients, adults or adolescents.
Great, thanks.
I think we are starting to run out of time here. Is there any other last issues people want to bring up? Something that hasn't been addressed? Anybody who hasn't spoken up yet wants to say something?
Not to beat the IL18 horse more to death, but I'm just interested because now that we're sort of measuring this, like Alexi's group, at diagnosis in almost all these patients, and I don't know how much of this is real or not, but it seems as though there's more similarity between two patients in whom the IL18 is really high even if one doesn't have arthritis, then between two patients who meet ILAR criteria, one in whom the 18 is normal and the other it's 40,000. In terms of how we treat them, their progression, I would say both groups seem to respond reasonably well to IL-1 and IL-6 inhibitors, but in terms of just likelihood of progression to arthritis or risk of MAS, they seem to be distinct. And I wonder if that's one of the things that has sunk that initial tadakinig trial in stills was all stills. And, you know, there really very few of those patients actually had really high IL-18 levels at enrollment.
There you go, Scott. I mean, in the end, IL-18 is molecularly defining a condition, and you can say it's molecular defining Still's disease, and in the future, maybe that's what we'll think of as Still's disease. But I don't know if it's really defining what we've called Still's disease or systemic JIA in mass. And I know that only 2 to 4% of patients in Boss's cohort have low IL-18, but I just worry that, you know, systemic JIA is going to become hyper IL-18, and then there's going to be, well, I guess there's another set for us to go study separately from the high IL-18 autoinflammatory systemic JIA. My concern is just without a gold standard, we're defining systemic JIA by IL-18 today, and I'm not sure that it's right. It very well might be. I'm just not sure. We don't have a way to really know.
Alexa, you're muted.
No, I just wanted to say that we do not define systemic JIA by the levels of IL-18, but we stratify those patients with systemic JIA based on the level of IL-18 and perhaps treat them differently. I think that's an issue.
Well, the other thing is that we do get some more, how do I say, some additional help in the differentials. If we have a patient that looks like systemic JIA and we get plenty of IL-18, then it's another, you know, piece of evidence adding up to the early diagnostic workup. And this, for sure, we all do that. I'm sure we all do that. And you do also, Michael.
Don't get me wrong. I'm not opposed
No.
to IL-18. My concern largely is just that this has the potential to lead us to using IL-18 in the diagnosis of systemic JIA. And
one of the pieces,
Yes,
not the
yes.
only one.
Yeah, I think you're totally right, Mike. Like, it's not like you can't get a horrendous chronic fever and a rash and cytopenias and organomegaly and have a normal IL-18 because we call it MISC and Kawasaki disease, right? So, you know, there's certainly a subset of patients, and you know, in Boss's group, it's a very small subset, which I feel like we'd have to look at our own data. I feel like it's certainly more than 2 or 4% for us.
We haven't looked. I will do that.
Well, it depends. It depends if you mean normal IL-18 or just not crazy high, because we don't really pay attention if it's less than 10,000.
I can't answer the question. I have to look at the data. Bas?
So Bas, could you comment on the 2 to 4% of your patients that didn't have IL-18? Was that didn't have it elevated at all, or just it wasn't highly elevated?
No, crazy high. I think he has a problem with less than 1,500. It's in the chat, less than 1,500. I think we have to homogenize.
Yes, so I just typed it in the chat, but for us, it's not crazy high and it's in the level.
You're frozen.
What is your upper limit, Boss, in your lab?
Yeah, I can show maybe some. Can you hear me?
We can hear you.
While we're waiting, does Dan have his hand raised? Oh.
Dan, yeah.
Yeah, so I have 2 comments. Nikolai, having worked with the clinical trials for decades now, the extrapolation of data from adults to children.
Yes, can you hear me?
With rheumatic diseases has worked well for those diseases in which the availability of adult patients and the size of the clinical databases are large enough to inform efficacy and safety before we do studies in
Okay,
children.
so the upper limit,
But with systemic JIA, the tables are turned to the opposite, where the size of the patients in the pediatric population is much more common, and trying to drive trial decisions based on adult data is going to very much delay the process. And it's not like we're going to have large databases of patients with adult onset Still's disease to inform safety and efficacy decisions about children. So I think generalization of that concept to systemic JIA is risky, as been stated before. The other thing I think we need to remember about diagnostic criteria and use of IL18 is that everybody in this room the clinicians come from a major academic center. And when you use diagnostic criteria, it needs to be applicable to all people who see patients with JIA, so that the diagnostic criteria for new onset patients, if it were to require IL18 as a criteria, would be very difficult to do in many parts of the world that don't have access to sophisticated labs like the people on this conference call do. So I think the diagnostic criteria using a variant of the clinical criteria and readily available standard laboratory tests is the way to go with diagnostic criteria, so that our colleagues in less privileged medical settings can still diagnose the disease and initiate treatment. So those are the only two comments I have.
Thanks, Dan. I just want to clarify that we certainly acknowledge the unique case of SJIA and adult onset Still's disease. And in this case, we actually extrapolated the other way around from pediatric with SJIA to adult onset Still's disease based on the same principles, but recognizing that SJIA is more prevalent in pediatric, Still's is more prevalent in pediatric patients. So we certainly recognize that the prospect of direct benefit principle applies to other conditions, not necessarily to SJIA. And I think we're open to continue thinking about this, the same way. Yeah. Regarding your second point, I'll leave it for the rest of the group if they have any follow-up.
Wow, everyone is quiet. I'll just make one point that I think about diagnosis that Randy made in the chat, that nobody's, it's not a question of requiring IL18 for diagnosis, but as a way for clinical trials. I think the diagnosis thing is a bigger question. It requires validation. There seems to be, willingness to look at that, but I think that's a bigger question. But I do think that we can come up with a pragmatic way to use something to be able to sort patients into the right trials. That should be doable even at this time point.
Well, just again, some regulatory perspective on IL18. Again, think about it as a biomarker, and biomarkers can have multiple uses or multiple roles in drug development. They don't necessarily need to be the defining factor for registrational trial or for confirmatory trial. They can have many other roles, and I don't know if Chris Leptic is still on the call, but he has certainly a broader perspective and can comment on that. So again, I just want to emphasize that this is not something that we would require, and I think it would be important to consider the generalizability of the findings if you define a population in a certain way, and that might be IL18, for example. And I think what would be actually more important is for us to know whether an intervention would also be effective in patients who are not, high IL18, right? there are ways that you can potentially enroll everyone and stratify based on IL18 level to get some more useful information on that. Again, these are some suggestions. I think we as an agency are certainly open to continue this discussion, but ultimately the role of IL18 would be determined by you and how you see its use.
Understood. Thank you. That's really helpful. So I think that, tomorrow we'll take some of what you're saying and what we've heard today to kind of come up with a few proposals for how we can actually, in the current time point, define a way for this group, subgroup, I would say, and come up with some trial ideas and continue this discussion, but maybe be able to get to some more concrete ideas. But I think you've given us a lot of room to, a lot of ideas to think about between today and tomorrow. On that note, we are a little bit over time, but it's been a great discussion. Are there any last comments? I mean, Scott, do you want to say something? Should we
I think today was super dense. I think that we made a lot of progress. I think that there's a lot of agreement among us and reassuring, input from Nikolai. I think that the hard work, if the goal is to bring trials to these patients who are not currently being served, the industry is the linchpin here. And so, I think that we can with this move forward to them with a sort of united front. But, I'm not good at predicting what their interest or response will be.
We don't know that, but at the same time, it's important to point out that when they direct trials at needs which are more met, then they have a very hard time recruiting, which is what we are seeing right now
sure.
in the trials in the SJIA space. And we face that all the time, is that, companies come to us and, we have a patient group, et cetera, and we point out how when you have a bunch of medicines and patients have a bunch of options, then, why your clinical trials will not be, enrolled in as well. So I think the problem does involve industry too. And we are hoping that, of course, we don't have any real say, to be honest. That's the dilemma, yeah. But at the same time, I do think several of them are in the call, and at least they are hearing us where we think as a group of physicians and parents where the unmet needs lie, So, okay.
But ultimately, the scientific questions are going to be asked by us as physicians and parents. So I guess if we have an agreement, then we know what we can do, what we have to do science-wise to keep things moving in the field, to make things moving in the field as fast as we can, Ash. And it's us who have to make it happen.
Sure.
Thanks.
Okay, well, on that note, we'll be here bright and early. I think I have to catch up with several of you later in the bit, Alexi, Scott, if you want to work on those proposals for tomorrow, but I'll ping you all by email. Thank you so much. We've had a maximum of 170 people join today, which is way more than I was expecting. So thanks everyone for joining, and we'll see you tomorrow. Thank you. Thank you, all the doctors. Thank you, Nicola. Thank you, all the parents. Bye. And thank you to our team, the MCI team. Thanks.